Hey everyone, what is happening? So we are getting into how nerve impulses are being passed from one neuron to another or even to an effector. All right, and so this is the title of this is Synaptic Cleft and Neurotransmitter Substances, and we've learned about how signals are propagated along a neuron. So more specifically, an axon. So this was the last video, and if you haven't checked that out, please do. That was with sodium and potassium, and what we're going to be doing today is learning about what happens when a neuron ends, okay? That message, that impulse has to be passed on to another cell, another neuron, or to an effector, which is a muscle or gland. Now, this happens at a relay point called a synapse, all right? So this is between an axon terminal of a sending neuron and the dendrite of a receiving neuron or an effector or muscle or gland. All right, and this whole thing is called a synapse, and this is what it looks like. So I'm going to draw it for you. All right, so what we have is we have our axon terminal, and this is an axon bulb. And so we've got our axon terminal there, and we've got postsynaptic cell. This will be either a dendrite, muscle, or a gland. And this little part right here in between, this is called our synaptic cleft, and it's about 50 nanometers thick, which is about 1,000th of hair. So the thickness of hair, it's 1/1000th, all right? And so this whole thing is called a synapse, all right? So we know that our impulse is moving along this way. It's being propagated, and we have sodium on the outside, and we have potassium on the inside. And we know that for an impulse to be passed along, we have to change the voltage of that neuron. And so sodium is traveling through voltage gated sodium channels, and then that's going inside the cell, and then potassium is being sent out by voltage gated potassium channels, and that's repolarization. And then we use sodium potassium pumps to get that back to what it used to be so that it's ready to be sent again. All right, so we know that from our last lesson. Now at the end of a axon terminal, we have voltage gated calcium channels. Now what happens is, as the presynaptic neuron is changing its potential, the voltage gated calcium channels, so we're going to put some calcium right here, are caused to be open. So as that we get depolarization and repolarization, it causes these calcium channels to open, and calcium rushes in. Now what do they affect? Well, we're going to put this in. So we've at the very end of our axon terminal, we've got the axon bulb. It's like that bulgy part, and we've got all these vesicles filled with a neurotransmitter substance, and we're going to get into the different types. But for this part right now, we're going to have acetylcholine in there, all right? And so what happens is we had sodium going in, we had potassium going out. That changed the potential of our neuron, and that change in potential caused our voltage-gated calcium channels to open. Calcium went in, and then now what happens is calcium rushes in and it binds with the vesicles, all right? So we get calcium binding with the vesicles that have the neurotransmitter substance in them. And what they do is they cause them to go and bind with the presynaptic membrane, all right? And so I'm going to draw a new picture here in a sec, and then they're going to empty their contents, all right? So what we have is we have our neuron being propagated, our nerve impulse being propagated, and the calcium moved in, and it caused the vesicles to go to the presynaptic cell membrane. And what happened was it released the contents into that synaptic cleft. All right, and it did that through exocytosis. And remember that from our cell membrane, and that's when things are getting sent out of the cell. And so it turns out that the neurotransmitter substance is going to bind to channels on the other side on our postsynaptic membrane. And so I'm just going to draw these in. Now, these channels look like this. And this is a cross-section one. And now there's this little spot on it where the neurotransmitter substance goes. All right. And so this membrane protein is going to allow for the basically the neurotransmitter substance goes in right here, and it causes the protein to open up. And what it allows in is more sodium to cause the impulse to be continued on. All right. So these are going to be some channels right here. They're all going to be opened up. And what they do is they cause sodium to move in, and then the impulse is moved along that way. All right. So then the neurotransmitter substance is either broken down within the synaptic cleft and it's reused, or it is sent back in and put back into vesicles. All right. Now this is really important so that the signals are brief and precise. All right. Now there are some neurotransmitter substances that can either make it excitatory, so it'll propagate that neuron or that nerve impulse, but some are inhibitory. And what they'll do is they'll decrease the signal propagation, and they don't allow sodium to be put in, and that's when it'll turn something off. All right. So we've got excitatory, and these increase signal propagation by opening sodium channels, and then that changes the potential, the electrical potential of the neuron, and that goes back to getting sodium in. It gets past that negative 55 millivolts threshold, and we get a rush of sodium in, and it causes depolarization, and then we get repolarization with sodium, and it just continues on just like we know. We also have inhibitory, and this decreases signal propagation. And what that does is it opens potassium channels or chlorine channels, which make it harder for the neuron to get past that threshold that we talked about in that last video. Now we've got a whole bunch of different neurotransmitter substances, but I'm going to just touch base on a few. And we've got acetylcholine, and this is released by motor neurons to activate skeletal muscles. And depending on what type of receptor it is, or what kind of receptor it is that it hits, this neurotransmitter substance, acetylcholine, it'll be excitatory or inhibitory. All right. So it makes skeletal muscles contract, but it actually slows the contraction of cardiac muscle, which is interesting. All right. And we have just a couple other ones. We have norepinephrine, which is an excitatory neurotransmitter substance. It's also a hormone, and it's going to help us get into that excited state to fight or flight. We also have, so we'll just put fight or flight. We also have serotonin, and serotonin is involved with sleeping, mood, attention, and learning. And this is also involved with, it's a precursor for melatonin. So melatonin, which we talked about, which is released by the pineal gland, which helps with sleep. Serotonin is the start of melatonin. We just add a few things to it and it becomes melatonin. All right. We also have dopamine, and you've probably heard about this. This is also dealing with sleep, mood, attention, and learning. Now, there's a couple disorders that we have that are involved with the lack of these neurotransmitter substances. Parkinson's is associated with a lack of dopamine in the brain. Oh, by the way, dopamine is also involved with pleasure. Then we've got schizophrenia. This is associated with an overactive dopamine neurotransmitter substance in the brain. And then we've also got depression, which is reduced levels of dopamine and serotonin. So just remember that for a neuron to be affected by a neurotransmitter substance, it has to have a receptor for that neurotransmitter substance. All right, so I'm going to show you a quick little picture of this. It's got some great steps on it. This whole lesson is going to lead us into, we're going to have a quiz on this, and but we're also going to lead into a little assignment about neurotoxins. All right, and so neurotoxins are things that are going to prevent neurotransmitter substances from propagating those neurons, and those can be really, really bad if you had like a cardiotoxin or something like that that actually stops the heart. All right, so I want you to check out this little picture, and this will be on the notes that we have associated with this lesson. All right, I hope you found this informative, and we will talk to you later.